I'd like to introduce our next speaker, and so my mistake on the order of presenters, but very much excited to introduce Akansha from UT Southwestern. Akansha will be sharing today work on the impact of distant tumor growth and the immune modulation on gut microbiome. So thank you so much for joining us today, Akansha. We're so looking forward to hearing about your research.
Thank you, Robbie, for the introduction. And before I start, I would like to thank Beyond Science Initiative Administrative Committee to give me the opportunity to present my work. Hi, everyone. I'm Akansha Singh. I'm a research track assistant professor at UT Southwestern Medical Center, Dallas. And my major focus is on key finding that some immunotherapies work in certain set of patients, but it doesn't work in others. So why this is happening? And this work is a part of that initiative. And we are trying to see how gut microbiome changes can impact the treatment outcome, or there was association vice versa here. So just to give a brief introduction about gut microbiome, we have millions of bacteria that are residing in our gut, and they play a crucial role in maintaining our metabolism and energy levels. And it helps contain the inflammation in the intestinal area. If there's a dysbiome, there's a change in balance of bacterial population. It causes chronic disease like irritable bowel syndrome. You guys must have heard about it. And as they say, we are what we eat. So researchers have shown that to be true, that there is a strong connection between our gut bacterial population and our mental well-being that can influence our mood and cognitive function. So as I'm a cancer immunologist, my focus is on how the tumor growth or the tumor immunomodulatory changes can be associated with gut microbiome. For that, I'll give you a brief overview what has been done till now. So there are many research where the cancer growth and its treatment are associated with gut microbiome dysbiosis. So there are certain type of bacteria that are beneficial for your gut health, and some that if grow in a certain number can cause inflammation. So these bacteria has been found to be associated with increase in tumor progression or metastasis, or can help immune cells to generate anti-tumor phenotype. So one of the example is Fusobacterium nucleotatum, which is very well studied bacteria to promote MDSCs and TEM accumulation in tumor microenvironment. It also binds directly to the tumor cells and help the tumor cells to escape T cell recognition and thus the cytotoxicity. On the other spectrum, the well-studied bacteria is Bifidobacterium, which is directly involved in dendritic cells activation and enhances T cell infiltration in tumor microenvironment. And there is an indirect correlation between bacteria and the gut microbiome changes, is that a metabolite that is released from these bacteria can modulate immune cells to differentiate into certain type of cells that can then again either promote the tumor cells or act as anti-tumor cells. So these bacteria, they come into different subsets. And it's not just the tumor growth that is directly involved. It's the treatment that we are giving to the patient that can influence our gut microbiome composition. And the two major treatment that are known to influence this is chemotherapy and immunotherapy. Like there are certain cells that are more sensitive to chemotherapeutic drugs than the others. There are bacteria that are more sensitive towards these chemotherapy drugs. And one of the example is lactobacillus, which is very important for our gut health, which get degenerated in the chemotherapy receiving patient, and they are given probiotic to help maintain their gut microbiome and healthy digestive system. On the other hand, if we talk in context of immunotherapy, there is a correlation between the responders and the non-responder based on the gut microbiome abundance of certain bacteria. So the bacteria that we just discussed, Bifidobacterium and Akkermansia, they are known to be, their higher abundance is known to be associated with good response in the patient against immunotherapies like anti-PD-1 or CTLA-4 checkpoint inhibitors. Now, the current focus of this microbiome research in cancer is associated with the tumors that are in close proximity or are associated with bacterial rich tissue. So the tissue like your gut, your lung, and vaginal region of females, they are in direct contact with the bacterial population. And the most of these studies that I have discussed were associated with the cancer happening in those regions. And also the treatment that are given intravenously, or there is a systemic treatment given to the patient, their correlation has been found with gut microbiome. So where basically these bacteria had the direct abundance, direct interaction with these drugs from the blood supplies happening in the intestine, and there is a clear correlation. But where our research come in is to see whether intratumoral treatment happening in the tumor that is far away from the gut, whether that treatment can cause some changes in gut microbiome, or there are gut microbiome features that can causes the immunomodulatory therapies to work at the distant site or not. So in the preclinical setup where we have a closed system, that is mice eating same kind of food and getting same are kept in same environment, we wanted to have a model which can act as a responder and not responders, and to naturally have those differences among the treatment population is to focus on the age and the sexual dimorphism, as it is known that females are more responsive towards the immunomodulatory therapies. So we looked at this impact of sexual dimorphism on gut microbiota. So it is a very simple experiment that we did. We collected the fecal sample from the male and female mice and then inoculated tumor in these mice. And after 20 days of tumor growth, we again collected the fecal sample and compared the 16S ribosome sequences to identify the bacteria and their abundance in those fecal samples. And what we observed is changes in alpha diversity majorly. So alpha diversity is the diversity of different population in the sample. So how many population of bacteria, how different bacteria are residing in your gut can be studied using alpha diversity indexes. So here I'm showing Simpson diversity index, and you can see very clearly the female healthy mice had more abundant, more different type of bacteria compared to the male. And with the tumor inoculation, there was no significant change in female alpha diversity, whereas in male, there was a significant change. So basically in males, there was a significant significant increase in the alpha diversity with tumor inoculation. Then we looked at the quantitative differences in the bacterial population at phylum level. And one of the major component that clinician look that work in this gut microbiome studies is the ratio of bacteroidetes to firmicutes. So based on that, they predict whether basically they analyze whether there's a dysbiosis happening or not. So the increase in firmicute is also associated with dysbiosis, and you can see in both female and the male with tumor inoculation, there is a significant increase in firmicutes population, which is in line with earlier studies that has been done in this context. Next, we wanted to see whether there is a bacterial population change that is associated with tumor growth. So as it has been shown earlier also, and we knew this before we started the study, that female mice will have a slower growth of tumor in comparison to male. So we looked at genus level how different bacterial population are changing, and we observed that there are some sex independent changes and some sex dependent changes. And sex dependent changes basically include Turicibacter, Streptococcus and Staphylococcus, which will play a significant role in our earlier experiments or outcome. Now, our next target was to see whether there is a impact of sexual dimorphism on immunomodulatory therapy outcome. And for that, we used two immunomodulatory therapies. First is calreticulin nanoparticle, and second is focused ultrasound-based histotripsy. So these treatments themselves are very different. So calreticulin nanoparticle are used to transfect cancer cells to upregulate calreticulin protein in those cells. And these were delivered in lipid nanoparticles. So because of lipid stress, this calreticulin get towards the surface and get exposed on the cancer cell. Now the macrophages and other APCs will see the calreticulin, which serve as eat me signal, and they will start phagocytosing tumor cells. On the other hand, the focus ultrasound mediated histotripsy treatment, it lyses the tumor mass. And the tumor lysis happen in a way that the proteins are not denatured because there is a millisecond heating, and the cell disrupt because of the sound wave, releasing all the tumor associated protein in the tumor microenvironment. And then again, the APCs can take up these tumor antigen and migrate towards the lymph node, tumor draining lymph node, and activate T cells. So we gave a total of 2 histotripsy treatment and 3 CRT treatment in different set of mice, male and female separately. And again, on day 20, we sacrificed these mice to analyze tumor cell infiltration in the tumor microenvironment that is growing in the subcutaneous flank region, and collect the fecal sample to see what is happening to the gut microbiome density. And what we observed is that in female, both the immunomodulated therapies, CRT nanoparticle and histotripsy, they showed a significant decline in tumor growth. But in male, histotripsy was not that significant, or it was not able to show any therapeutic efficacy as it showed in female. Whereas CRT nanoparticle showed similar kind of tumor delay as it showed in females. Next, we wanted to see whether there is a specific type of immune cells that is accumulating in the tumor microenvironment before we start comparing it with the gut microbiome population. So we did a comprehensive immune analysis using flow cytometry, and we looked at different cells, T cells, CD3 positive T cells, and then NK cells, as well as APCs like dendritic cells and macrophages. And what we found out that CRT treatment, it enhances APC's activity. It polarizes macrophages towards M1 type, and there was higher accumulation of T helper cells in tumor microenvironment. Whereas with histotripsy treatment, NK cells were upregulated. And these responses are very specific, are more prominent, I could say, in females rather than males. So why NK cells came with histotripsy is because NK cells are the responder cells to any kind of injury happening in the tissue. And H2 treatment is kind of a thermal injury that we give to the tumor samples. So NK cell presence was not something predicted here. And all of this activation of either APC or NK cells lead to the effector T cells to accumulate the tumor microenvironment, which we confirmed by the interferon gamma expression and granzyme B expression on these CD8 T cells. And again, this response was more prominent in females compared to the males. Now, we looked at the association of these therapeutic efficacy of treatment and the immune cell infiltration in the tumor microenvironment with the gut microbiome abundances. So what we observed that at phylum level, as there was the increase in firmicutes, in females, this firmicute level decreases further beyond the healthy mice composition, which was again not seen in males. Therefore, we correlated it with the less extent of immune cells changing in the tumor microenvironment. And there were a certain bacterial genus that were abundant in certain type of treatment groups, and our focus was more on the non-responder group, that was histotrypsy treated males. And we observed that there are 3 or 4 microbiome population that were more abundant or was significantly accumulated only in these mice, and 2 of them were Enterorhabdus and Streptococcus. Here we are just showing 15 top bacterial population that has changed, but overall there were 31 genuses that were found with left C analysis, and we will be comparing those next with the immune signatures in the tumor microenvironment. Now, this 31 bacterial population, when correlated with tumor volume, immune cell infiltrating the TME, and serum cytokine level, there were many that showed significant population with 1 or 2 immune signatures. But we wanted to focus on the bacteria that are very strongly associated with either with the tumor volume or with the immune signature changing with tumor growth or the treatment in these mice. So based on those, because this heat map is kind of not very clean here, so I put the concluding remark on the side. So 4 genuses, Enterorhabdus, Subdulogrelum, Desulfovibrio, and Sacrobacter, they showed a positive relation with tumor growth. Basically, the mice where the tumor was growing faster, these bacteria were more abundant. Whereas the mice where the tumor was smaller and the tumor was growing slowly, Turicibacter and Peptococcus were more concentrated in the fecal samples. Other than these 6 bacteria that showed direct correlation with tumor volume, there were bacteria that showed a very strong correlation either with this immunosuppressive cells or the protumoral cytokines, or a set of antitumoral cytokines as well as cytotoxic T cells. These were Streptococcus, Staphylococcus, Gastrophilus, and Rhombosia. So based on these observation in this study, what we concluded that first, to start with, a healthy female has more alpha diversity and male has less alpha diversity. In the clinic, it has been observed that patients with more alpha diversity are known to respond better to an immunotherapy than to the patient who have lesser alpha diversity. So we can conclude that female there could be an immunomodulatory association here. And once the tumors start to grow, the treatment is given to those mice. So there were few bacterial population that were highly associated with responders with increased cytotoxic T cell and antitumoral cytokine levels in the serum, and those were Gastrophilus, Rhombosia, Turicibacter, and Peptococcus. Whereas there were set of bacteria that were associated with non-responders. Out of these 4, Desulfovibrio associated strongly with the non-responder profile and higher immunosuppression profile in the mice. So, okay, we got these results, and there has been study that has shown earlier that there is a correlation change that gut microbiome changes has with cancer development or the treatment outcome. But what is doing what? So for the future research, we need to develop a causal relationship. We have to understand definitely that whether a tumor growth is changing gut microbiome or certain gut microbiome are promoting tumor cells. That will help us define the therapeutic interventions, like whether if it is the bacteria that is changing the tumor response, then we can focus on fecal matter transfer like therapies. But if it is just the immunomodulation happening because of the tumor and its treatment, which is changing gut microbiome composition, then we can use those bacteria to predict whether this patient will respond to the immunotherapy, certain type of immunotherapy or not. And at last, we have to validate all these findings in a clinical setup, whether what we are seeing here is actually patient appropriate or not. With this, I'll conclude my talk here, and I would like to thank my faculty mentor Ashish Ranjan for providing me with the resources to conduct this research, and my lab members that has worked with me at different time point. And in last, Beyond Science Initiative, thank you guys for giving me the opportunity to present my work here.